In eukaryotic organisms, there are two types of cells, somatic cells and germ cells.
Somatic cells, or body cells, are exact diploid clones of the parent cell, and they are created by mitosis.
But germ cells, or gametes, are haploid cells created by meiosis.
Let's say we are watching meiosis in Drosophila melanogaster, or fruit flies.
The parent cell starts out as diploid in interphase, meaning it has two sets of chromosomes.
Its DNA is in a loose form called chromatin in the nucleus, so that it can be transcribed and duplicated during the normal life of the cell.
During the synthesis phase of interphase, the DNA is replicated so that the parent cell now has 4 sets of chromosomes, tetraploid.
As the cell prepares to enter the first stage of meiosis, prophase 1, the chromatin wraps itself tightly around the histones into chromosomes, and the nuclear envelope breaks down.
Each chromosome is made up of 2 genetically identical sister chromatids.
The chromosomes pair up with their homologous pair, which has different versions of the same genes. Each homologue comes from one of the parents.
The homologues form a tetrad and switch random homologous portions of their genetic material. This is called crossing over, and it increases genetic variation in the resulting gametes.
Once the homologues have swapped genes, the cell enters metaphase 1. The chromosomes independently assort themselves along the cell's metaphase plate, and the spindle fibers attach to the chromosome's centromeres.
Independent assortment during metaphase 1 also increases genetic variation.
In anaphase 1, the spindle fibers pull the chromosomes apart. We should end up with 2 equal clusters of chromosomes on either side of the cell, as long as non-disjunction does not occur, but we'll get to that later.
In telophase 1, 2 new nuclei form around each cluster of chromosomes. The chromosomes unwind back into chromatin, and the cell finally undergoes cytokinesis.
So now there are 2 diploid daughter cells entering meiosis 2. In fruit flies, that means there are 8 chromosomes in each cell.
Here we go again. Each daughter cell enters prophase 2, and the DNA condenses back into chromosomes, and the nuclear envelope breaks down.
In metaphase 2, the chromosomes line up along the metaphase plate, and the spindle fibers attach to the centromeres of every chromosome.
The spindles pull the sister chromatids apart in anaphase 2. They pull half the chromosomes to each side of the cell as long as non-disjunction does not occur.
In telophase 2, 2 new nuclei form at both ends of the cell, and the chromosomes become chromatin again. The cell splits into 2 haploid daughter cells, which each have half a set of chromosomes.
So let's add everything up. We went from 1 tetraploid parent cell to 4 haploid daughter cells.
Right, and because a fruit fly's diploid number is 8 chromosomes, then we split 16 chromosomes 4 ways. That means each haploid gamete has 4 chromosomes.
In male fruit flies, there would be 4 sperm, and in female fruit flies, there would be 3 polar bodies and 1 egg.
The random combination of 1 egg and sperm creates even more genetic variation. Genetic variation is important because it increases the likelihood that some organism will have the right combination of genes to survive.
After fertilization, 2 haploid cells form 1 normal diploid cell again.
If non-disjunction does not occur.
Let's rewind back to anaphase 1.
What if the spindle fiber doesn't connect correctly to the centromere of one of the chromosomes?
That means by the end of meiosis, 2 of the daughter cells will have 1 extra chromosome, and the other 2 will be missing a chromosome.
This could also happen in anaphase 2. If 2 of the sister chromatids aren't properly pulled apart, then 1 of the 4 daughter cells will have an extra chromosome, and 1 will be missing a chromosome.
Non-disjunction in both anaphase 1 and 2 is a major issue because an extra chromosome will cause overexpression of those genes, and a missing chromosome will keep the cell from producing enough of the necessary proteins.
Fetal growth and development is such a precise process that trisomy and monosomy are usually fatal. And in the rare cases that an organism survives, there will be severe birth defects.
Although you probably aren't familiar with common non-disjunction related conditions in fruit flies, you've probably heard of many in humans.
Both Down syndrome and Edwards syndrome are human genetic diseases caused by trisomy.
The results are catastrophic when meiosis goes wrong, but fortunately, this very precise process rarely makes errors.
Meiosis must be the most efficient process yet to evolve amongst eukaryotes because it is conserved between species as disparate as fruit flies and humans.